Hello, welcome to this video. So this is biochemistry or chemistry 3111. So in this video we are going to talk about glycolysis, the metabolism of monosaccharides, disaccharides and the polysaccharides. So specifically for this introduction, we are going to talk about catabolism of monosaccharides. How are these monosaccharides broken down exactly? So let's dive into that. So monosaccharides are broken down using a common pathway. And this common pathway is called glycolysis. So the common pathway is called the glycolysis. So glycolysis, we can write it as these are just a series of reactions that involve conversion of glucose, which is a monosaccharide, especially in animals, humans. So this glucose is converted pyruvate pyruvate and a trace amount of ATP. Yeah. So glycolysis is just a series of reactions that convert glucose to pyruvate and a small amount of ATP. So where does the glycolysis happen exactly? So this one happens in cytosol of the cell. In cytosol. So what's the importance of glycolysis? Importance of glycolysis. So one, glycolysis is important because it degrades glucose to pyruvate. So degrades glucose to pyruvate. And secondly, glycolysis is important because it provides raw materials that are needed for other reactions. Provides building blocks or raw materials. It's one and the same thing. for synthetic reactions. So it's synthetic reactions. These are just reactions that involve making some other molecules, like for the formation of fatty acids. So let's now dive into the pathway exactly for glycolysis. Glycolysis pathway. So for the glycolysis pathway, since we are breaking down glucose, and the ultimate results of this pathway is the formation of pyruvate. So first and foremost, you're going to have glucose. It's the first thing. We have glucose. Sorry, glucose. So this glucose has to be converted, gets converted to glucose 6 phosphate. So here you use energy. There is need for energy, ATP. So that means you need the phosphate. So since in ATP you have 3 phosphates, so this one you are going to get 1 phosphate from ATP. So when you get 1 phosphate here, you are going to add it to glucose. And when you remove 1 phosphate from this ATP, that means this ATP will be converted to ADP, meaning you have 2 phosphates. So this one here, you have the first enzyme now, the one that catabolizes this reaction. And this enzyme is called hexokinase. Hexokinase. Remember, glucose has got 6 carbons, hence hexokinase. So after that, this glucose 6 phosphate has to be converted again in a reversible. And remember, this first reaction here for the conversion of glucose to glucose 6 phosphate, this reaction here is irreversible. That means that when you convert glucose to glucose 6 phosphate, you can't go back. So let's go now. We continue with the converting this glucose 6 phosphate. So this glucose 6 phosphate, okay, let's use a white color so that, so this reaction, it's reversible. So that means it can go back. It can go forward and backward. So this glucose 6 phosphate is converted to fructose 6 phosphate. Six phosphate. So this reaction, as you can see, fructose 6 phosphate. So here the enzyme that is involved is in phosphoglucose isomerase. The enzyme is phosphoglucose isomerase. So let's use a different color here. Let's use like a blue. The enzyme will be, so this will be enzyme number 2. That will be phosphoglucose isomerase. Yeah, so glucose 6 phosphate is converted to fructose 6 phosphate using phosphoglucose isomerase. Thereafter, this fructose 6 phosphate now gets to be converted again. It's converted to, so here, this one, when it's converted here, that means there is no going back here. Once it's converted, it goes to the glycolysis. So meaning this one is the one that we are going to call the committed step. So this means that glucose is committed to the glycolysis process. So this one will be converted to fructose fructose 1,6 by phosphate. So here the enzyme, now since we are talking about the committing step, so the important enzyme that is involved here is the PFK, which is phosphofructokinase. So this is the third enzyme, phosphofructokinase. And this is what we call PFK1. So let's put it in brackets. So here let me emphasize a certain point here. So here, as you can see, so this is one of the most important enzyme. Let me even write it in red so that you don't forget. Important enzyme. So this one is the one that we call the committed step. I mean the limiting step. Limiting step. Where this fructose 6 phosphate is converted to fructose 1,6 biphosphate. So like, why is this step exactly a limiting step? So it's because this step here, it's irreversible under cellular conditions. Let me just write it somewhere. So one, irreversible. Under cellular conditions. Then number two, it's highly regulated. Or controlled. Then number three, it commits glucose to the glycolytic pathway. Yeah, so I've explained now why this step here is called the limiting step. So make sure you understand the limiting step. So after you form fructose 1,6 biphosphate, the next step involves, so the next step, so here there is also need for converting ATP. So ATP here is converted to ADP. Yeah, so this is another point here. ATP is converted to ADP since the phosphate is the one that is added to fructose 6, so that you have fructose 1,6 biphosphate. So that means to this fructose here you have two phosphates. So the other step now, this is the other step. So this one will be pointing backward. So here you have fructose 1,6 biphosphate. So this one is converted to dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. So here there is one thing that is most interesting that we need to understand here. Let's draw it properly. So we have dihydroxy acetone phosphate, then here we have glyceraldehyde 3 phosphate. Phosphate. So here there is need for this one here, dihydroxyacetone phosphate. This one has to be converted to another glyceraldehyde 3 phosphate, because this one cannot be used in the glycolysis. So this means that you need an enzyme here, and this enzyme here, it's see, so the reaction for here to be a reversible reaction. It can go forward and backward. So there we go. So the enzyme here is going to be triose phosphate isomerase. So remember, dihydroxyacetone phosphate and glyceraldehyde phosphate, these are 3 carbon compound. So we have triose. Okay, let's use another color here. Let's use yellow. Triose phosphate isomerase. Triose phosphate isomerase. So when this one is converted to another glyceraldehyde 3 phosphate, so that means you are going to have 3 of them, which is what is required for the glycolysis pathway to continue. So let's go on. Let's go here now. So another point I forgot here is the enzyme that is involved here. Conversion of fructose 1,6 biphosphate to glyceraldehyde and dihydroxyacetone. So the enzyme here, let's write it in red, is aldolase. Yeah, so this is the enzyme involved here. So after that, we have glyceraldehyde 3-phosphate getting converted in a reversible reaction, another reversible reaction. So this is a reversible reaction. You can go forward and backward. So the enzyme that will convert this one to, so 1,3 phosphoglycerate, 1,3 phosphoglycerate. So the enzyme that will be involved here is glyceraldehyde 3-phosphate dehydrogenase. Oh, let's use another color. Let's use yellow. Yeah, so this is the enzyme that will be involved here. So here there is conversion of NAD plus so this one is reduced to NADH, then plus protons are reduced here. So this one is converted to this one. Then again this one gets to be converted. So this one is also a reversible reaction here. So this one is converted to 3-phosphoglycerate. Phosphoglycerate. So here the enzyme will be phosphoglycerate kinase. Phosphoglycerate kinase. So there is conversion of ATP to ATP, ADP to ATP. So you've seen. So here there is formation of ATP. So this is what we call by substrate level phosphorylation, because there is formation of ATP from this substrate. Since 1,3 phosphoglycerate is just a substrate in the reaction. So let's write it in yellow color so that you can remember. So substrate level phosphorylation. Make sure you remember this point. So this is substrate level phosphorylation because ATP is formed from the substrate. ADP is converted to ATP using a phosphate that is gotten from this substrate. So another point. So after 3-phosphoglycerate, it's converted to 2-phosphoglycerate. Again, this reaction will be a reversible one. So 2-phosphoglycerate. So the enzyme here will be phosphoglycerate mutase. Let's use a yellow color. Phosphoglycerate mutase. The next 2-phosphoglycerate gets to be converted now to phosphoenolpyruvate. So this one involves hydration. So water is given out. So the enzyme here that converts 2-phosphoglycerate to phosphoenolpyruvate, it's enolase. Phosphoenolpyruvate. Enolase. Oh, let's use a different color so that we can see it clearly. So this is phosphoenolpyruvate. Then for the enzyme, it's enolase. So this phosphoenolpyruvate gets to be converted to pyruvate now. Which is the end product that we needed for glycolysis. So here the enzyme will be pyruvate kinase. So here we remove phosphate from this one here. So that means we are going to have ADP get it converted to ATP. So this is another substrate level phosphorylation. So wherever there is formation of ATP, so that's what we call substrate level phosphorylation, which are the 2 steps that we've seen so far. Thank you for watching the video. Make sure you subscribe to the channel as we make more videos for chemistry 3111.